We are now ready to start talking about concept 2 for our cells, and this is going to be talking about cell transport. So how things move in and out of our cells. So the goal of cellular transport is always going to be homeostasis. And homeostasis is the need of an organism to stay stable by regulating its internal conditions. This is always the goal at a cellular level and at the entire organism. So we call this a dynamic equilibrium, and this is maintained, but it isn't always the same, but it's going to stay within some range. So an example of this is meaning dynamic, so it can move. We have our pH scale, and so we're going to always be on some range of what is acceptable for an acidity level or basic level. We'll be at some range for body temperature, ranging from hot to cold. We know that our body temperature likes to be around 98.6, but sometimes it's a little higher, sometimes it's a little lower, and same with blood sugar is another good example of this. So as an organism, we are constantly taking in stimuli and have to respond to these stimuli in order to maintain homeostasis. So if we look at this, a stimulus is a change in our environment, and the response is a change in the organism as a result of a stimulus. So as we're maintaining homeostasis, we will often see what are called feedback mechanisms. And these mechanisms evolve to help maintain homeostasis in organisms as we respond to stimuli. So there's a variety of different ones of these, but the mechanisms use the output of a system to signal a change in input so that a system can be either stabilized or amplified. So these are really the two things that we're going to look at. Are we going to stay the same, or are we going to do more of something? So we will always have our input, then something in the system happening with our output. And they can either be positive interactions or negative ones. So first we're going to look at an example of a positive feedback mechanism. And in a positive feedback loop, the output or the product of a system is always going to intensify the response. So we're going to go over some examples of this. But the first one is human childbirth. And in this, these are supposed to be little arrows here. But we have hormones that are going to cause contractions. These contractions are going to cause an increase in pressure. This increase in pressure is going to cause release of more hormones, which causes more contractions and more pressure. So by amplification, we're meaning we're getting more and more and more of something. A second example of positive feedback mechanisms is fruit ripening. So it's the same sort of thing as our childbirth, that as fruit slowly ripens, it's going to release this enzyme, which is then going to signal more fruit ripening, which causes neighboring fruit to ripen, and then more release of this enzyme. So it's important to remember that positive feedback intensifies the response, so it's amplifying it. We're getting more and more and more. This is different from our negative feedback mechanisms. So in a negative feedback loop, the output or product of a system causes a counter response or a return to a set point. So we're trying to go back around in the loop. So we're going to call this a stabilization mechanism. So some examples here is our human body regulating its body temperature. Another example in humans is water concentration regulation. So think about how dehydrated you are is maintained by negative feedback loops, and then our blood sugar regulation. All where we are trying to go back around to get us where we want to be. And as we talk about maintaining this type of homeostasis, we really need to focus on the cell membrane. So homeostasis is maintained through the regulation at the organ system level all the way down to the cellular level, which is what we are focusing on now. So most of homeostasis on our cellular level is maintained by the cell membrane, meaning that our cell membrane is going to be controlling what moves in and what moves out of the cell. When talking about the cell membrane, we will often say that the cell membrane is selectively permeable, which means that it is picky. It is picky about what goes in and out of the cell. So first, let's talk about the things that can pass easily through the cell, things that the cell membrane likes. And these are things that are small, non-polar, hydrophobic, and or neutral, as well as water. So kind of weird to think about, but our non-polar hydrophobic things, so oil, water-fearing things, or neutral things like water can pass easily through the cell. Whereas things that cannot pass easily are things that are polar or very large molecules. They will have more difficulty passing through. As we talk about cells moving through the cell membrane, our transport of materials into and out of the cell can be classified in one of two ways, as either being passive or active transport. So today we're going to briefly talk about each one, starting with passive transport. And passive requires no extra energy by the cell because molecules are moving from high concentration, meaning they're all squished together, to areas of low concentration, meaning they're spread out. So this is going to be going down the concentration gradient. So I like to think of this as going down a slipper slide. It doesn't take very much energy for you to go down the slide, which means it's passive. There's no energy being used. So examples of this are simple diffusion, facilitated diffusion, and osmosis. Then we have active transport, which does require extra energy in the form of ATP to be spent to bring materials into or expel materials out of the cell, and we're moving from low to high concentration, which means we are going against the concentration gradient. So instead of from low to high concentration, now we're moving uphill. So you're going from low to high, which means you're going to be climbing up the slide, which does require this extra energy. So we have molecular pumps, exocytosis, and endocytosis as our examples. And before we move on today, we need to remind ourselves of some of these important words we have discussed. So the word solute, if we remember, is what gets dissolved. So we can think back to our lemonade powder. This is what gets dissolved. Our solvent does the dissolving, and if we recall, the universal solvent is water. And then we have the solution, and if we add these two things together, we get the solution. So the solution is a uniform mixture of two or more substances, like the lemonade that we just made. But we're adding a new term today, and that is concentration. And concentration is the amount of solute that gets dissolved in the solvent. And we like to abbreviate this with these little bracket signs when we don't want to write out this big long word. So a way to remember concentration is something you already know. If we're making lemonade, maybe you like strong lemonade or weak lemonade, and you change how strong it is based on the amount of the lemonade powder that you add, the amount of the solute.